Well, let's get going. So last time we'd gotten to the point of RNA editing, and I'll try to summarize what we talked about RNA editing last time on a slide at the beginning. But most of what we're going to talk about today is protein synthesis. But the two lectures are in fact linked because the RNA, which has been synthesized, the mRNA, which has been synthesized in the nucleus, must leave the nucleus and go out into the cytoplasm to provide the substrate for protein synthesis. Now, we're going to find that regardless of what kind of RNA we have, whether it's messenger RNA, transfer RNA, or ribosomal RNA, all of these RNAs must be transcribed from DNA in the nucleus. Once they're transcribed from DNA in the nucleus, however, their fate is to leave the nucleus through the nuclear pores, enter the cytoplasm, and in the cytoplasm begin the process of protein synthesis. These three kinds of RNA all work one with the other. They all work in concert to make proteins. And briefly, the mRNA, messenger RNA, encodes for the amino acid sequence. And it encodes for the amino acid sequence through a 3-base set called a codon. Now, remember there are 4 bases in the RNA. If each base corresponded to an amino acid, we would be stuck in a world with 4 amino acids. If we had 4 bases that we could use 2 of them interchangeably back and forth, if you look at how you can rearrange 4 base sets into different combinations, you get 16. Well, we don't live in a world of 16 amino acids. Maybe there's another place that does that, but we don't do that here. We have 20 amino acids. So using a 2-base set on the mRNA isn't sufficient to account for the total number of amino acids we have in our biological molecules. That means we have to use a 3-base set. The 3-base set is the codon. If you do the statistics on that, you see that a 3-base set has 64 different combinations. So we have 64 different combinations for 20 amino acids. So there's certain redundancy in the system. It's called the degeneracy of the code. Now, that's a detail. But what I do want you to hold on to is that there are going to be 3-base sets called codons in the mRNA that are going to determine the amino acid sequence of the resulting protein. But the mRNA in itself is not sufficient to make proteins. It has to work in concert with 2 other kinds of RNA. One of the other kinds of RNA is called transfer RNA. We abbreviate this tRNA. The transfer RNAs carry amino acids to the ribosomes. The transfer RNAs are like shuttlecraft. But there are more than 20 different transfer RNAs because we have this degeneracy of the code. But each one of the transfer RNAs will covalently link to a specific amino acid. We'll see that there are 2 main characteristics of the transfer RNAs. First is that they have an anticodon. They'll have a 3-base set called an anticodon that can match up with the codon in mRNA. The other major idea of the transfer RNA is that the 3 bases in the anticodon will determine which amino acid is going to be covalently linked to the transfer RNA so they can be a shuttlecraft to bring these amino acids into the ribosome. So mRNA determines the amino acid sequence. The transfer RNA determines what amino acids are brought into the ribosome. And the transfer RNA contains information about which specific amino acid a given tRNA will carry. Finally, the ribosomal subunits, the small and large ribosomal subunits, are made predominantly of ribosomal RNA. Now, the ribosomes are made up by about 60% ribosomal RNA. The remaining 40% is proteins. So you have to have the proteins. But what I want to emphasize here is the importance of the ribosomal RNA in constructing this factory, the ribosome, which is now going to form the peptide bonds. As we work through this today, what we'll see is we have to have all of these 3 working together. The mRNA will be sandwiched between the large and small ribosomal subunits. The large and small ribosomal subunits will move along the mRNA in 3 base sets called codons. And as we progress down the mRNA from the 5' to the 3' end, as we move along the mRNA, the transfer RNAs are going to be the shuttlecraft bringing specific amino acids in. The specific amino acid that will be deposited will depend upon the anticodon-codon pairing. Then the large ribosomal subunit has two main effects on this. One is it will cut the amino acid loose from its attachment to the transfer RNA and form a peptide bond. The second one is that it forms a peptide bond. So what we'll see then is the enzymatic activity of the ribosome is to cut an amino acid off of the transfer RNA and anneal it to the peptide chain, which is growing on another transfer RNA, and we'll walk our way through that. This idea of 3-base set is really fundamental. The idea of having a 3-base set, and each 3-base set determines an amino acid. That then determines the amino acid sequence. These 3-base sets known as codons, I'll never ask you to memorize. There are way too many of them, and any cell biology book you ever look at will have a table someplace, or you can look it up on Google to find out what the 3-base sets are for each amino acid. So I'm not going to delve into that, but there's one I want you to know. The one I want you to know is AUG, adenine, uracil, guanine. AUG is in the mRNA, and it always encodes for the amino acid methionine. But in addition to encoding for the amino acid methionine, that's the begin here sequence. All proteins start with an mRNA sequence of AUG that determines a methionine as the first amino acid. That sets the reading frame. That AUG says we're now going to read every 3 bases from there on. And as you read every 3 bases from there on, each one of those 3 base sets will determine an amino acid until you get to one of these 3 base sets. The 3 base set of UAG, UGA, and UAA are said to be the stop codons. When you encounter one of these 3 base sets, you've come to the end of the mRNA, you've come to the end of the reading frame. And the transfer RNA that is complementary to these so-called stop codons doesn't carry an amino acid. It carries a termination factor. And that says that's the end of the protein. The protein comes loose, we've generated a brand new protein, the large and ribosomal subunits come off the mRNA, we'll find another mRNA, and the process begins again. Just a word on this. I mean, again, I won't ask you to memorize this, but it's sort of interesting. They all begin with uracil, and then the other 2 bases are always a purine, either adenine or guanine. The one that's missing here is UGG, and that encodes for tryptophan. So again, we're going to walk along the mRNA 3 bases at a time. We're going to match the codon with the anticodon, and then we're going to form a peptide bond. And that's fundamentally what we're going to spend most of our time on today. But in this process, if we are generating a protein on a ribosome that's free in the cytoplasm, that will now generate a cytoplasmic protein. But that opens the question, how do we use this machinery to make a transmembrane protein, a lysosomal protein, or a secreted protein? How do we manage to target the machinery to the endoplasmic reticulum to make rough ER? And the answer to that question lies in a series of amino acids, initially at the amino terminus of the protein as it's being made. This is called a signal peptide. It's at the N terminal. The signal peptide, if the sequence of amino acids out here is predominantly hydrophobic, that's a signal peptide that now says, I'm going to bind to a signal recognition particle. If the hydrophobic amino acids of the signal peptide bind to the hydrophobic amino acids in the signal recognition particle, that's a signal to take the entire machine, the mRNA and the ribosomes, to the endoplasmic reticulum. In the endoplasmic reticulum, there will be a receptor for the ribosome, and there'll be a receptor for the signal recognition particle. At that point, there will be a signal to disassociate the signal recognition particle from the signal peptide. And then the hydrophobic amino acids in the signal peptide will be handed off to a transmembrane protein called a translocon. The translocon is a transmembrane protein in the end of and then protein synthesis will be again, and we'll deposit the protein in the ER. We probably won't get all the way through this today, but I'd like to get as far as we can. And then finally, we'll see a sequence of amino acids within the protein that are hydrophobic. They too will bind to signal recognition particles, but within the protein itself, within the string, these now become the start and stop sequences for transmembrane proteins. So we'll try to round all that out. But let's start here with the slide. And this is where we left off. And this is a little bit more complicated, but not a whole lot. This band up here is meant to represent the pre-RNA. This is the band of messenger RNA that is absolutely complementary to the DNA strand from which it was transcribed. So every base in this RNA has a complementary base in the DNA. And if we were to simply be talking about prokaryotes, that would be the end of the story. We'd make a protein from that mRNA. But eukaryotes have this clever trick. And the clever trick that we closed on last time was that there are enzymes in the nucleus. There are nuclear enzymes that will cut out some segments and tie other segments back together again. Now, what we're showing here in blue are the so-called exons. These are the regions of the mRNA that will use the bases in that region to be converted into protein. So this is going to be the edited transcript down here. But then these yellow regions are the regions now that will be cut out and discarded. Those are the so-called introns. So introns are the intervening sequences that are discarded. The exons are the regions that will be expressed as protein. Now, why this is important is that most organisms contain about 20,000 genes. This was a surprise from about 15 or 20 years ago. We thought that most animals would have much, much larger genomes than that. But we have a finite number of genes to make a much larger number of proteins. So how did that get done? Well, some of that results because of the variability in the splicing at these points. So the enzymes that are causing these cuts and resplicing, those enzymes are recognizing base sequences in the mRNA. And we have a whole host of these different enzymes. And if you put your phone away, I would appreciate it. And so those host of enzymes then will find slightly different places here in the pre-mRNA to make the cuts. So if you imagine at this interface here, between this little snippet out at the end and this intron, if we moved the cut point a little bit to the right, now we'd include a little bit more of this intron with that exon. So that would make a longer region in the mRNA with bases from which we could make an amino acid sequence. So by altering just a little bit the places where we can use the enzyme to make these cuts, we can change the base sequence and the resulting edited mRNA. That will change the amino acid sequence and the resulting protein. What this means then is we can use this finite 20,000 number for the genes and make a much larger number of proteins from this so-called alternate splicing, how we splice these together. Some proteins will be arranged in such a way this whole intron will now become part of an exon. You just don't make it, you just read right through it and make an exon out of it. So this provides a mechanism to make slightly different proteins, a large number of slightly different proteins from an individual mRNA molecule. So any questions on that?
What are
yes?
the modifications that can make a protein that's not harmful?
So the question here is, can you make a mistake and make a protein which is meant to be not a problem? And that's probably true. I don't know of any specific cases where that happens, but if you have a change in a mutation in the DNA, which now changes the sequence of bases in the RNA, which now changes the splice site, you can now make another protein that may not fold properly. So there are possibilities for that. Yes, and then yes, okay?
I'm a little confused as to why the splicing needs to happen and why you can't just read the whole thing. Like, why is there a reason to splice it?
So why do we bother to splice? Well, because why questions are always difficult, because we describe what's happened, but why it's happened is really difficult to know. But having said that, pretty much when you generate this, there's no way coming from the DNA to the RNA that we can change that sequence very well. Biology just didn't do it that way. So you make an RNA transcript that's complementary to the DNA strand. So then the next step is you're going to have to modify the RNA. No. You're going to have to modify the RNA so you get a different base sequence so that will give rise to a different protein. Then subsequently, we can modify the proteins. We'll see later we can phosphorylate them and tyrosylate them and do a whole bunch of things. But at this point, all we can do is read the DNA to RNA and then change the sequence in the RNA. And the way we change the sequence in the RNA is to simply cut out some sections and splice them back together again. And the way the enzymes work, you always have to anneal together these adjacent regions. You can't cut them apart, for example, and if this is 1 and this is 4, you can't cut out 1 and anneal it to 4. You can only cut out 1 and anneal it to 2. And it's the way the enzymes work. And if you get into this, there's something called a lariat formation. They actually make a loop, and part of the loop is to cut it and splice it back together again. And that loop formation is why you can only stitch together adjacent exons. And you had a question.
Is the exon downstream from the start codon, is that like the promoter region, or because what's the purpose of that exon if the start codon is where?
So the question here is, why do we have this bit up here if the start codon is going to be downstream a bit? And it looks like many of these proteins will have large regions that won't be translated into proteins. These are untranslated regions, both at the 5' and the 3' ends. And it appears that why that is, I don't know. But there appear to be often quite large stretches that the small ribosomal subunit we'll see in a moment will get on to that end, or get on to that end of the mRNA, the 5' end. If you put your phone away to appreciate it, it will move then please put your phone away, from the 5' to the 3' end until it encounters the start codon. Yes, in the back.
With RNA splicing, do you just arrive at messenger RNA, or do we get transfer and ribosomal RNA as well?
So this is a question about RNA splicing and how universal is it? We usually tell this story from the point of view of the messenger RNA because that's where it's most common. But having said that, there is some splicing that also occurs, particularly in the ribosomal RNAs and to some extent in the transfer RNAs. But it's just not nearly as extreme. Anything else? Yes?
How do the enzymes know which portions will become introns and which portions will become exons for that specific protein?
So so this gets back to a little bit more depth that I wanted to deal with. But how we know what will become an intron and what will become an exon has to do with the way the enzymes come along and read the bases in the mRNA. They will come along to a consensus site, and they'll read, I think it's 12 to 15, something in that neighborhood of bases. And they'll say, I'm going to bind here. And then they'll recognize another set downstream someplace. And they'll also have another set of bases, because these enzymes also contain some RNA in them. And so they'll base pair downstream a little bit. And when they base pair downstream, then that determines the other end of the intron. That point, those two are pulled together into a lariat, into a lasso. And then at that point, the loop is cut out, and the places where the lasso, the lariat comes back together again, those are annealed together. So you can get variations in the length of the intron depending on how the enzyme is recognizing the bases in the RNA. And by having slightly different base sets, you can move it one way or the other. Okay? Yes?
Are the introns just disposed in
the introns just go away. They don't even have to go to a lysosome. I don't think they have to go to lysosomes. I think there are enzymes, there are endonucleases in the nucleus which will cut them down to their appropriate bases. Then they have to be recharged with ATP and all that. Anything else? If not, let's now start here on this panel and describe protein synthesis, whose other name, and I'll put this in parentheses, translation. on ribosomes. Now again, the three players in this story will be mRNA, transfer RNA, and ribosomal RNA. Well, the mRNA is simply what we described in that slide. This will be the edited mRNA. I should add too, sort of a diagrammatic relationship of where we are. Remember, if this is the nucleus, this is the site of all transcription. But you'll remember from our early discussions of the cell that out here in the cytoplasm are ribosomes. So in fact, we can point out here the important concept that all three of these RNAs, which have been synthesized in the nucleus, these all function in the cytoplasm. This mRNA is like any of the mRNAs we've seen. It has a 5' end, and it has a 3' end. And then all along its length is the string of bases. And I'll just put some of these in at random, where this is a uracil. I better be careful. A uracil, a guanine, and a guanine. We'll do that one. And an adenine, and a uracil, a cytosine, and then we can do an adenine, a uracil, and a guanine, and then we can do U, and say a G, A, C. Now, the small ribosomal subunit will now get on to this 5' end of the mRNA. So this is the small ribosomal subunit. Let me label this one as the mRNA. This is edited mRNA. Now, the small ribosomal subunit will walk along. It will walk along the mRNA from the 5' to 3' end. So it will move in this direction. And it will move down the RNA strand until it reaches this 3 base set called the start codon. The arrival of the small ribosomal subunit at this 3 base set recruits a transfer RNA. Now, a word on transfer RNAs. Transfer RNAs have this interesting shape. Some people call it a sort of cloverleaf pattern. This end is the 3' end. The 3' end is the end to which we can bind an amino acid. The chemistry of this is such that there are enzymes that will recognize this transfer RNA and covalently link an amino acid to this 3' end of the transfer RNA. But the other major idea we need to hold on to from this generalized figure of a transfer RNA is that down here at this end, in purple, is a 3 base set. This 3 base set has a name. It's called an anti-codon. And the anti-codon base pairs with the codon in mRNA. Now, as we said before, there are some 64 different possibilities of the codons and a similar number of anticodons. But each anticodon will determine what amino acid that transfer RNA is going to carry. That determination of what amino acid this transfer RNA is going to carry is made by a cytoplasmic enzyme called an amino acyl transferase. What that enzyme does, what that cytoplasmic enzyme does, is it finds the anticodon. It recognizes the anticodon, and it will be carrying a specific amino acid that is appropriate for that anticodon. So in this case, we're going to see for the start codon that if this is the anticodon for the tRNA, the anticodon will be adenine to uracil, uracil to adenine, guanine to cytosine. So the anticodon is UAC. Anytime a transfer RNA is carrying a UAC anticodon, it's going to be carrying out here methionine. Because one of these amino acyl transferases will have recognized this transfer RNA with its UAC anticodon and appropriately linked a methionine to it. Then the arrival here at this site of the transfer RNA with the methionine, along with a small ribosomal subunit, in some way that I don't understand, recruits now this other character, which is the large ribosomal subunit. Now, it's true that both the small and large ribosomal subunits contain a significant complement of proteins. About 40% of these ribosomal subunits are proteins, but the remaining 60% is ribosomal RNA. And that plays an important role in the enzymatic activity of these subunits. So, the next thing that happens here is that we've now set this arrangement up so that the large ribosomal subunit comes in and finds the small ribosomal subunit and this start codon with its anticodon attached to the transfer RNA. If you stare at this for a little bit, you'll begin to recognize that what this does by staying here, we're going to start with this 3-base set of AUG. That now sets it up so we can read 3 base sets down the line. That's said to set the open reading frame. Because if you think about it for a minute, you recognize the only way we can make this thing function is to read 3 bases at a time. We have to read them in 3 base sets. And if we're going to read them in 3 base sets, there must be some way of saying each subsequent 3 base set is going to be for an amino acid. The way we determine that every subsequent 3 base set is for an amino acid is going to be by setting the reading frame here with the UAC and the start codon. Yes? So the question here of the ribosomal subunits. These ribosomal subunits always move from 5' to 3'. We won't get into the details of this, but normally this is actually sort of a circular structure, and up here there'll be a 5' cap. There's a 5', there's a GTP cap on this, the 5' end, and that's an important signal to tell the ribosomes to start here. That's an important signal to tell the small ribosomal subunit to get onto that end. And they'll always walk from the 5' end toward the 3' end. And in most of these, if you may have seen this in some other classes, the 3' end will have what they call a poly-A tail. You have a whole string of A's at the end of it. And that has to do with telling this thing to come to an end. But they always read 5' to 3'. And that usually that poly-A tail will then form a circle by attaching to the GTP cap. Yes? So how does the large ribosomal subunit get there? It appears to be something to do with the transfer RNA and the small ribosomal subunit. But exactly how that gets done is not clear to me. I don't know if it's known. It's probably known, but I don't know it. And yes?
So do all proteins then start with methionine?
So the question is, do all proteins start with methionine? The synthesis of all proteins starts with methionine. This is true even in prokaryotes. This is called an F-met methionine, slightly modified. It's a formal methionine, F-met. It has a formal group attached to it, but that's a detail. But we always start the synthesis with the methionine, but by the time we make these proteins, we usually cut those methionines off. So not all proteins have their initial amino acid methionine. Yes, and then we'll go back there.
Is anything happening to the codons prior to the start codon?
So does anything happen to the codons prior to start codon? No, there's often a large untranslated region out here that can be quite several bases long that seem to be just filler and may be a site to help organize the small ribosomal subunit as it reads along. Sometimes it will miss one of these AUGs. There are sometimes a few of these AUGs along the line to say start here. And there was another question in the back. So the question here is there only one small ribosomal subunit? There's only one small ribosomal subunit for each large ribosomal subunit. But if you were to take the cytoplasm here and grind it up and purify small and large ribosomal subunits, there may be more or less an equal number, but there will be hundreds and hundreds of thousands of small ribosomal subunits and thousands of large ribosomal subunits. So I don't mean to imply there's only one of these even for every mRNA. If you get down into the weeds of this, you'll find that typically an mRNA will have multiple ribosomes on it simultaneously reading along that mRNA, simultaneously making protein. Right. Better move on. So now, the AUG sets up reading everything in 3 base sets subsequently. Now, I've deliberately chosen U here because I know that there will be an anticodon of adenine, adenine, adenine. And I know, because that's an easy one to remember, that this is going to be carrying a phenylalanine. Now, let me put the phenylalanine on the other side here because it's a little easier to think about. So this is the phenylalanine that's linked to the 3' end of the transfer RNA. This is the acyl transfer RNA. Now, what will happen when these two are lined up is that the large ribosomal subunit will do 2 things. It will cut loose this methionine from its acyl bond to the transfer RNA and now form a new bond between the methionine and the phenylalanine called a peptide bond. And in fact, the 3-dimensional structure of the RNA is what is catalyzing that enzymatic reaction. It's one of these old arrangements where the RNA itself is catalytic. And the catalytic activity of the ribosome is twofold. It will cut loose the amino acid on this transfer RNA and link it to the amino acid attached to this RNA. Some of you may have seen this as the amino acyl site and the peptidyl site in some of your other classes. That's what this is. This is just a fancy way of, that's a fancy way of determining that you have 2 sites bound. You have 2 sites for the transfer RNAs. And in one of them, you have only the acyl, and the other, you have the growing polypeptide chain. So then, this whole machine moves over. Let's put some more RNA in here. Let me just do a CAGs with me. So now the entire machine moves down 3. So I'll put this in sort of a dotted line. So now we have this growing polypeptide chain, attached to the transfer RNA that was carrying the phenylalanine. Now, the GAC here is going to have an anticodon, and this anticodon will be CUG. Now, I have to look at this and see if I actually put one in here that I have on my list. GAC. I don't have it. Okay, so there's an amino acid here. That amino acid will be specific to this GUC anticodon. I don't know what that is. Go to a table, you'll find it. But now, because this whole thing has moved over, and now we have these two sites occupied, what will happen is the ribosomal subunit now will cut loose this amino acid and attach it to this phenylalanine. And then that will now release this guy, release this tRNA, and the entire machine moves down again. We do this over and over again so that we now have a polypeptide chain being added to this last transfer RNA that was moved into the site. And so we do this over and over again. So now there's going to be a GUC with its, and CAG is, I think that's glutamine. So this is now going to be glutamine. Again, what we'll do is we'll clip this glutamine loose and attach it to this growing polypeptide chain. So one at a time, we're adding amino acids, making this chain longer and longer and longer. And so as we do this, this is going to be the amino terminus, and we're working our way toward the carboxy terminus. We do this over and over again until at some point we reach a 3-base set, which could be UAG, or it could be UGA, or it could be UAA. But now we do have a transfer RNA that has an anticodon for this. So there's this guy that's carrying this structure. And this will have an anticodon of adenine, uracil, cytosine. But out here on the 3' end, it's going to be carrying a termination factor. And the termination factor can't be added to the growing polypeptide chain. But yet the ribosome still cuts loose the growing polypeptide chain from its attachment to the tRNA. So now we have liberated this sequence of amino acids as a protein. And we can't attach to the termination factor, so that says that's the end of the protein. So again, we start here with an AUG start codon. That sets the reading frame to be thread 1 codon at a time, or 3 bases at a time. And as we walk along, each of those bases, because of the codon-anticodon interaction, will determine the amino acid that will be added as we synthesize the protein until we reach the AUG stop codon, which can't form a peptide bond, and that's the end of the protein. Yes, you had a question. I thought you had a question.
I was just a little unclear. You said that some proteins will keep the methionine attached and some will just cut it loose. So if they do keep the methionine, will they instead add on the methionine to create the chain, or will the methionine be added onto the second one only?
So the question here is about the methionine. And as far as I know, all the time we're synthesizing these proteins, it will contain a methionine. I don't think until the protein is finally translated that methionine is clipped off. And yes?
How fast does this happen?
How fast does this go? I think each of these readings occurs in like a microsecond. That may be fast. But it takes seconds to make a protein. It doesn't take hours. And so you'll read along these or at least minutes. You'll read along these, and these can be, you can have 400 or 500 amino acids easily. So that's 1,200 to 1,500 bases, and those all have to be read. And I think those are running in microsecond time scale, certainly not second time scale. So reasonably fast. Yes?
After the RNA on with the AUV, does the RNA automatically create peptide bonds?
So the question here is, do we automatically, once we've cut this methionine, basically it happens in reverse order. The methionine in this case will be peptide bond linked to the phenylalanine, then this will be cut loose.
Does it have to be a UUU or could it be?
Oh, it could be anything. No, I'm sorry. I just put it in the UUU because I knew that's phenylalanine and I could remember it. And I wasn't smart enough to remember GAC. So that's just an amino acid. But it could be any triplet. Although it's not going to be much of a protein if the next triplet's a stop codon. But I would like you to see, though, that all we have to do in here is take one of these out. Take one of these guys out. That's a point mutation in the DNA. And then this system doesn't know any better. It's going to start with a start codon, and it's going to read along here. And because now we're going to get a frame shift, it won't be very long until you run into one of these 3 stop codons and you prematurely truncate the protein. Yes?
So is it amino-acetyltransferase that puts the tRNA with the amino acid to the A site, and then peptidyl-transferase that makes the peptide chain?
So let's not get too into this nomenclature. Let's hold on to the idea that the amino-acetyltransferase works in the cytoplasm simply to add the amino acid. And then down here in the ribosome is where we're cutting loose the acyl bond and forming a peptide bond. And that's done by the ribosomal enzymatic activity. Let's leave it at that and not get into peptidyls and such. Anything else? Okay. In that case, let's take the last few minutes and talk about synthesis of secreted lysosomal and transmembrane proteins. You'll recognize this as proteins synthesized. These are proteins synthesized on rough endoplasmic reticulum. The process that I described above, and I should label this process is for cytoplasmic proteins. Using this simple model of our cell, there would be the large ribosomal subunit and the small ribosomal subunit. And as the protein was generated out of the large ribosomal subunit, it would fold into its 3-dimensional shape. And then eventually, when we came to the stop codon, we'd move that protein out into the cytoplasm, and we have a cytoplasmic protein. That's what we're talking about here. But that, of course, opens the question, how do we generate this other family of proteins? And we start the same way. We start with the mRNA. We start at the 5' end. We read toward the 3' end. We have then the sequence of bases here. We again will find the A, put it up here. We'll again find the AUG start codon. Well, that will help assemble the small ribosomal subunit and the large ribosomal subunit. So everything up to now is the same. This will then continue translating, moving along the mRNA 3 bases at a time, adding 1 amino acid for each of those 3 bases, just as we described above. As we generate the protein, and I'll try to show this in green, coming out of the top hat here, coming out of the top of the top hat of the large ribosomal subunit is the sequence of amino acids. This is the amino terminus. Now, if these amino acids coming out of the top hat are predominantly hydrophilic, we go the route that we showed up here on the cell. This protein is just dumped into the cytoplasm. But if these amino acids are predominantly hydrophobic, something else happens. So if this sequence of amino acids, usually in the neighborhood of about, I think it's 14 to 20 amino acids, if these are predominantly hydrophobic amino acids, if they're predominantly hydrophobic, then they have these R groups that are uncharged. And it's not a good thermodynamic state to have these uncharged amino acids out here in the aqueous environment of the cell. So what happens is there's another character. There's a character that looks a little bit like a guppy. And this character that looks like a guppy is said to be a signal recognition particle. It has this peculiar name of a particle because there's some RNA in it. It's one of these peculiar regulatory RNAs, but don't worry about that. This is a signal recognition particle, and what it recognizes is this sequence of predominantly hydrophobic amino acids, which is termed a signal peptide. So the purple guppy recognizes the signal peptide. So in green up here is the signal peptide. And in purple is the signal recognition particle. Now, if we cut a section through this guppy, we find that if we cut a section through the tail and we look at it end on, it looks like a half pipe. And at the bottom of that half pipe is a sequence of amino acids which are predominantly hydrophobic amino acids. That now allows for the hydrophobic amino acids of the signal peptide to rest here in the half pipe and through hydrophobic bonding help hide the hydrophobic side chains, the hydrophobic R groups on the signal peptide from the water environment. So we basically nestle these hydrophobic amino acids down in the hydrophobic region of the signal recognition particle. Then once we've done that, once these two are nestled together, I'm going to have to make myself some room here. Then once those are nestled together, the entire machine, the small ribosomal subunit, the large ribosomal subunit, and the mRNA, all of that find their way to the structure I'm going to show here in a box, being the endoplasmic reticulum. So on the endoplasmic reticulum, there will be this sort of structure which is an SRP receptor. It appears too that there will be a receptor here for the ribosome. Such that now the top hat here with its associated small ribosomal subunit and mRNA settle into this site. So the ribosome attaches, the ribosomal receptor binds to the ribosome. But let me finish this and I'll go as fast as I can. Then out here with the signal peptide attached to the guppy, what will happen is that some GTP will come along here, and that will separate the SRP from the guppy, and there will be a transmembrane protein across the phospholipid bilayer called a translocon. And now what will happen is the large ribosomal subunit will hand off the hydrophobic amino acids that were attached to the guppy. Those are now handed off to the translocon. So hold on to that idea till next time, and next time we'll start synthesis again, and we'll feed that protein into the lumen of the ER. It doesn't coil. It appears to, as far as I know, it doesn't coil. It seems to be like an